talk about the mark on methylcytosine and also post-translational modifications that are carried out on proteins, particularly on histones, altering their interaction with DNA and altering DNA structure. So I believe it was Sunny after class who came up to me and asked me, how are epigenetic marks inherited across generations? We talked a little bit about that, that epigenetic changes are made in one organism and then seem to be passed from one generation to another. Still a little bit controversial. But what's the molecular explanation of that? And it's a bit vague. It's kind of mysterious. We can appreciate that a chemical change on a DNA, such as methylcytosine, could end up getting passed along from one cell to a daughter cell. Probably be in a semi-conservative way, but it's possible to consider how that individual mark could then be propagated onto the other strand. And in that sense, we can kind of understand how in a simple organism, from one cell generation to another, epigenetic marks could be segregated. But in humans, across generations, this is a lot more difficult to understand what might be the molecular mechanisms. And our Danny Reinberg, who's actually a researcher here at NYU's medical school, says, in mammals, epigenetic marks are erased during the first two phases of the life cycle, first just after fertilization, and then in the developing primordial germ cells. So it's kind of mysterious. Like, we can understand that the cells may divide, but the retention of these chemical changes is still not clear. And then when we get to issues of histones, this also seems a bit peculiar. Okay, a cell can divide, and then some of the histones will be passed, presumably again in a semi-conservative way, to one daughter cell, and then the cell presumably will make additional histones. But this has been the focus of research from Danny Reinberg's group, and they've worked on model systems, and their early work suggested that modifications of new histones are patterned on those of the old histones, and that the new and old histones randomly assort between the two daughter DNA strands. So this maybe begins to suggest a way that we could make chemical marks on these histone proteins, and that these could be retained from one generation to the other. But I think Sunny's question was a really good one, because this is the cartoon that's shown in the book regarding inheritance of epigenetic states. And for me, when I see a cartoon that's this vague, it just makes me a little bit uncomfortable. Like, do you guys really know what's going on? So there's something like an activator protein which seems to get passed on from one generation to the next as the cell divides, but this truly is a bit mysterious. And this maintenance of epigenetic states, this cartoon is also a bit unclear. Okay, an epigenetic mark is established at a particular point in the genome, and then somehow the gene that's regulated at that site is able to maintain that state. Again, quite vague, but these are points which should, I think, not frustrate you, but should excite you, because there's so much to learn about how this might be carried out. And again, a lot of the interesting work is actually being done here in New York, much of it in the lab of Danny Reinberg. And they're trying to figure out how histone dimers might be replicated semi-conservatively. And when I see that term, semi-conservatively, it makes me wonder, it's like, could you test this kind of segregation by density labeling experiments? So that's kind of an interesting notion. But back in 2010, when they were thinking about these issues of epigenetic marks in histones, Professor Reinberg's comment was that only the methylation of DNA has been shown to be stably inherited between cell divisions. Now, he's just come out with a paper this year that is actually beginning to pry out a little bit more information about how this might be taking place. And what they're finding is that for repressive changes, chemical marks on histones, these post-translational modifications may end up getting inherited in an interesting way, in that if there are histones that are partially included with these post-translational modifications, if only some of the histones are, that could actually serve as a site for some of these enzymes to come along and recognize those modified histones and then pass along those same types of modifications onto neighboring histones on other nucleosomes that are close by. So this would involve these systems having both a reader that would recognize these post-translational modifications and an associated writer which would be able to carry out similar post-translational modifications in the area. So far, the evidence from the Reinberg lab seems to indicate that this kind of process can happen for these repressive states, but they don't have much evidence that similar things are happening in these active states. So again, this is just a little bit of a peek at what these sorts of processes might be that could lead to the inheritance of these epigenetic states. Overall, I think there's a lot of questionable science getting presented. I kind of brought this up last time. These issues about whether or not epigenetic marks that are associated with trauma could be passed from one generation to the other has also been a focus of research right here in New York. And the comment that's been made by John Greeley at the Einstein School of Medicine is that it's really difficult when you see an epigenetic mark, when you see a cytosine being methylated. Is that because there's been some event that's created that mark and then that's been propagated from one generation to the next? Or has this individual just obtained a gene sequence that is more susceptible to receiving these epigenetic marks? So is it an external cause, or is it due to the underlying genetic sequence? We just don't have the tools right now to be able to discriminate between those two possibilities. And the comment that he's made, I think, is really very insightful. That's the problem with epigenetics. The stories get out of hand because they're such wonderful concepts. If we allow the Deepak Chopras of this world to take it over, we're going to destroy the brand of epigenetics, and no one is going to take even the good science seriously. We have to be really cautious in the kinds of stories that we tell. And even when we have very compelling scientific hypotheses that really appeal to us, we have to make sure that we have the data to back them up. All right. So let's talk about gene therapies. What I think is really interesting, I began the semester talking about our ability to develop personalized medicine and understanding that perhaps with respect to cancer diagnosis, that these diseases are heterogeneous. They arise for different reasons and they need to be treated in different ways. So if we can obtain genetic information about the expression of particular genes, or the presence of particular mutations, that's really going to guide our ability to fine-tune treatment for that particular patient. But instead of observing some type of disease state associated with poor gene expression, deleterious gene expression, and then trying to provide some type of drug medication to compensate for that, increasingly, many of you are going to have an opportunity to actually manipulate those genes themselves. So if there's undesirable gene expression, you will have the tools to shut down the expression of those particular genes, or perhaps manipulate the sequences of those genes themselves in your patients. So how can we begin to shut down gene expression if that's associated with a particular disease state? One technology that's been around actually for quite some time is that of antisense oligonucleotides. So here's the DNA associated with a particular gene. We're going to be creating an RNA that's templated off the antisense strand, right? So here's the mRNA that's going to be encoding our protein. And if we were to design a synthetic sequence that's complementary to that mRNA, all we would need to do is just to synthesize 5' to 3' that antisense strand. We can do that either with DNA or RNA. Presumably we would prefer to work with DNA because of its enhanced stability. So what could we do with that? Well, once that gene was transcribed into mRNA, if we had the antisense oligo, we could use that to complex with the mRNA within the cell, within the cytoplasm, and prevent that mRNA from being translated. How might that occur? Well, this could block translation directly because this double-stranded mRNA wouldn't be compatible with ribosomal synthesis. But in addition to that kind of steric blockade, the formation of these types of hybrid structures actually can engage nuclease degradation of the mRNA. Okay, so normally we would generate an mRNA from a gene that would lead to a protein. If we come in with an antisense oligonucleotide, that hybrid structure is a substrate for a ribonuclease called ribonuclease H, which cleaves the mRNA and prevents that protein from being expressed. So that's a great way to shut down the expression of that deleterious gene. And this just shows you a little bit about the general activity of the ribonuclease H, which is endogenous within the cell, and this is the structure of that enzyme. So taking hybrid DNA, RNA, and then cleaving the RNA substrate. The difficulty with implementing this in a practical way in the clinic is that, yeah, you can administer an RNA or DNA oligonucleotide, but the pharmacological properties are really, really poor. Nucleases are present. We've talked about the intrinsic hydrolysis rate of oligonucleotides. It just won't stay around very long, so they're very susceptible to degradation. Also, if we just administer oligonucleotides, we're actually going to be generating an immune response. Our bodies will recognize those as being foreign nucleic acid sequences, and we'll get an immune response. And then administering it to the body is one thing, but actually getting these molecules to go where they need to go is a major impediment. Realize that oligo-RNAs, oligo-DNAs, these are polyanionic species, and trying to get them to cross the cell membrane is going to be quite difficult. So even if you administer these things to a patient, the possibility that they'll actually be able to have an effect in the cytoplasm is pretty small. Nevertheless, there has been some therapeutic success with regard to these types of molecules. And this is one that's used to treat cytomegalovirus retinitis in the eye. It's kind of interesting to think about why the eye might be a good compartment for this type of pharmacological intervention. And this is a pretty interesting therapeutic molecule. And I won't belabor this point right now, but you might want to take a look at this and look at the differences in the chemical composition, particularly in the backbone of this molecule. You'll see that it really doesn't resemble typical DNA very much at all, and it's been modified in the backbone to make it resistant to nuclease degradation. So this is in use right now, antisense molecule that complexes with the mRNA, and it's clinically approved. I think there's a lot more enthusiasm about developing a new strategy that is associated with RNA interference. And this might have been somewhat exotic 10 or 15 years ago, but is now fairly commonplace to use in a research setting to provide for gene knockdown. And it's become common enough that I wouldn't be surprised if some of you have used it in laboratory research. Has anybody used RNA interference? Yes, one person? Okay, great. Fairly easy to implement. If you want to reduce the expression of a particular gene, you can add a piece of double-stranded RNA and effectively knock down the expression of that gene. So how does that happen? Double-stranded RNA is brought into the cell. Either this could be exogenous, or in the case of microRNAs, many of these are actually synthesized as gene products. Single-stranded RNA is taken to the RISC complex, and it's associated with target mRNA, and then it's cleaved into segments. And I'll have more details on this in upcoming slides. As some of you were walking in, I was showing a video from Nature that goes into some of the details and has a beautiful animation about how this process happens. I'll provide a link to that video, and that'll be a really great way for you to review this process as well. Your textbook has a description of RNA interference in section 21.4, if you want to review this. And there's two real main aspects of this that we need to know, both RNA interference and also the influence of microRNAs. So in the case of RNA interference, this we typically associate with the introduction of foreign, exogenous, double-stranded RNA. And there's a variety of ways to transfect this double-stranded RNA into a cell. Once it ends up in the cell, it becomes associated with a protein complex called dicer. It takes a longer double-stranded RNA and cleaves it, I believe it's into 21 to 23 nucleotide base pairs, is the size of the typical siRNA, small interfering RNA molecules. Once dicer has got it in this appropriate form, it transfers it to another protein complex called RISC, which loads one of the two strands into the RISC complex, and then uses that as a template to go out and seek an mRNA to destroy. So this is our opportunity to target a particular gene associated with a particular mRNA for destruction. What we need to be able to do is introduce a double-stranded RNA that's complementary to that mRNA, and that will be used by the RISC complex, which is capable of carrying out RNA cleavage. And that will target that particular mRNA for degradation, and the outcome of that is selective gene silencing. And we now have a lot of rules about what would be the optimal sequence of our mRNA to design a small interfering RNA oligo in order to maximize that gene knockdown. Okay. So a very similar process occurs in the miRNA pathway. These are RNA molecules that are actually synthesized as gene products in the cell. And these are used very often as regulatory elements. They're part of gene regulation that would take place in a normal cell process. In this case, the precursor RNA is cleaved, usually into a hairpin structure by a protein called drosha, and then the same downstream events happen. That's taken to the dicer complex, that's turned into the appropriately sized double-stranded RNA loaded onto the RISC complex, where it's used as a template to identify the proper mRNA species, and in this case, that either provides for a translational blockade or it can lead to the targeted degradation of that mRNA by RISC. the outcome is the same. We've selected a particular mRNA that we want to control its expression, and we've been able to knock it down. This is the link to the animation if you want to review it at some point. So this is now gone beyond just an interesting laboratory phenomenon, like, okay, I want to study the influence of this particular gene, what happens when I knock it down. Okay, now I can study the phenotype in cell tissue culture. This is now turning out to be a promising candidate for the development of therapeutics. Once again, if we have a gene that's causing some type of disease state, if we were able to go into the body, use these types of RNA oligonucleotides, knock down the expression of that gene, potentially we could treat the disease. And some of the first articles that were talking about the possibility of therapeutic silencing of endogenous genes came out maybe in the early 2000s, the early knots around 2004. It very quickly ran into severe problems with regard to implementing it in a clinical setting. And some of these we've already talked about with regard to antisense oligonucleotides. Sometimes it's not really about the drug, it's about the drug delivery. And that's the case here. If we can get these oligonucleotides into a cell, generally they're able to carry out their effects fairly well, although there can be some problems with what are called off-target effects when mRNA that's not being directly targeted also ends up getting caught up and degraded. But the main source of the problem is we're asking this therapeutic regimen to do a lot. Okay, we can create our therapeutic oligonucleotides, but here's what that material needs to be able to do. We need to be able to introduce it into the circulatory system. It needs to go through the circulation and end up in the target tissue. We need to make sure that it doesn't get excreted through regular metabolism, like renal clearance, for example. It needs to avoid being degraded. We already talked about that being a problem. There's nucleases everywhere. It needs to avoid immune recognition. It needs to be moved from the circulatory system and moved into the target tissue. That's a process called extravasation. And then it needs to get taken up into the cell, huge barrier there. And once it's present in the cell in an endosome, it needs to be released into the endosome so it can be engaged by the RISC complex. So that is a lot. Nevertheless, some of the first clinical efforts in developing RNA oligonucleotides as therapeutics actually just used bare, naked RNA in an effort to cure disease. A lot of those early clinical efforts didn't go too well because the RNA just didn't stick around too long. A lot of effort now has come about to develop new strategies for delivery of nucleic acids. Here are 3 different strategies. One is to encase these oligonucleotides in lipid nanoparticles. Others are to make chemical modifications to the oligonucleotides where we coat them with saccharide groups to protect them from nuclease digestion. And then there are some really interesting kind of cocktails where there's a combination of chemical modification of the oligonucleotides where we conjugate things like steroid groups, cholesterol onto them to make them more compatible with the cell membrane. And then we encase that into a polymer or a peptide, which is capable of transiently causing permeability of the cell membrane. So all these strategies have been tried, and very excited to say that there's been some really interesting clinical trials. Yeah?
Have you ever studied how viruses detect RNA as a way to develop strategy?
Yeah, there are a lot of viral delivery strategies for a lot of these genetic therapies. I won't really go into too much detail about that, but yeah, that also is another strategy that's actually more important than talking about CRISPR, which I'll get to in a bit. So a lot of these use these synthetic siRNA oligonucleotides and have different ways of formulating them or conjugating them so that they will be taken up into target cells. It's interesting to see what proteins what genes are being targeted for knockdown. In some cases, these are protein folding disease or protein deposition disease. And the company Alnylam has been going after an amyloid disease with the very simple idea, as we were talking about protein folding diseases before, like if you've got a protein that's undergoing misfolding, creating these extended beta sheet structures, getting deposited in the body and causing all kinds of problems with regard to protein deposition, just shut off the expression of that gene. And that may end up alleviating the problem. So that's what Alnylam has been doing. And some of the early results were not too encouraging. So just a couple of years ago, a press release came out where Alnylam said that, well, they had an imbalance of mortality in their clinical trial. It's like, wow, imbalance in mortality. What does that mean? It means, unfortunately, that they ended up killing a bunch of patients. So 18 people ended up dying in a phase 3 study. And this was just a complete disaster. This should never, ever happen. Their early phase 1, phase 2 clinical trials should really spot whether or not there's safety concerns that would lead to this kind of outcome. So this was a huge setback. There's a lot of discussion just a couple of years ago whether this was going to be the death knell for this type of genetic therapy and the use of siRNA. But they stuck with it. They tried reformulating the therapeutic RNA molecules. And actually, just a few months ago, the FDA has approved Alnylam to treat this transthyretin-mediated amyloidosis using an RNA interference approach. So there's a lot of encouragement here. I think they still need to find out exactly what types of genes, what types of protein products are going to be most amenable to this kind of therapy. But this is encouraging as far as the development of a completely new way of treating disease. Here at NYU, there's early research going on in a kind of an interesting avenue of this type of therapy, and that's to treat problems with vocal fold injury, which is a surprisingly common problem, not just for singers. And the question is how to deliver this siRNA oligonucleotides to the vocal folds. And this is work that's being done by Professor Ryan Bransky up at the medical school. We provided some compounds to them to complex with those molecules, including a polycationic site to complex with the DNA and a lipid type entity to provide membrane permeability. And that work there has shown that in the presence of the appropriate RNA oligonucleotides that are corresponding to a gene that causes scar tissue formation in the vocal fold, that they can actually knock down quite effectively the expression of that protein in vocal fold tissue. So those sorts of results are really encouraging, and many of you may have the opportunity to use these types of new therapies for gene knockdown. But there's even more powerful technologies that are on the horizon. And I'm really encouraged about what the next 10 to 20 years are going to provide as far as ending genetic diseases. So this is Jennifer Doudna. She is at UC Berkeley receiving an award with another researcher in the area of CRISPR-Cas machinery. Emmanuel Charpentier from France was another awardee. And this was a quite glamorous award event, recognizing the incredible contributions that they've made towards advancing the science of CRISPR therapies. So this is an approach for actual gene editing. So not just going and attacking a gene by blocking the mRNA, but actually figuring out ways to remove deleterious genes or change the sequence of deleterious genes. This has been a long-standing effort to provide for gene editing through a variety of different techniques. These are some of the older approaches that have been used in the past. Some of these have been using nucleases that actually recognize very extended DNA targets, as opposed to the typical restriction endonucleases that you might have used in the lab, which recognize only 4 to 6 oligonucleotides typically, and don't provide the kind of specificity that would be required. What you want to be able to do is create a double-stranded break in the DNA, right where you want to introduce your changes. And so other approaches have used zinc finger nucleases or an assembly of different protein structures, each of which can recognize a particular base pair, and tied these to nuclease proteins to introduce double-strand breaks at those particular positions. And these have led to some advances in a laboratory setting, but nothing with the kind of efficiency that would really make anybody encouraged to think that they could introduce it in the clinic. Now, however, we have this new technology called CRISPR-Cas9 that is also capable of causing these double-stranded breaks. And in some cases, what that will lead to is just the joining up of those breaks in such a way that we disrupt the gene. But if we also use that break as an opportunity to sneak in a new piece of DNA, we can actually use homology-directed repair at these double-strand breaks to precisely edit the DNA and insert alterations of genes or correct errors. And now you've got to be able to think like, hey, if I know that there's a particular mutation in a gene for hemoglobin that's causing sickle cell anemia, why don't we go into that patient and just fix that mutation so they're no longer producing that mutant protein? If we know a particular mutation in a transporter that's causing cystic fibrosis, let's not worry about taking care of the downstream effects of that faulty protein. Let's just go into that gene and correct that mutation in all the cells in the body and alleviate that disease state. So that's the basic premise behind being able to make these alterations in DNA and introduce this capacity for editing. So what is CRISPR? Does anybody have an idea of what this whole system is? It's gotten a lot of play in the news. I'm just wondering if anybody's familiar with it. Okay, so CRISPR is a gene editing technology, and it's composed of an endonuclease protein, right? So an enzyme that can cleave DNA, that is capable of being targeted to very specific DNA sequences, which is absolutely crucial. And that activity, that nuclease activity, can be directed by a short sequence of RNA that's complementary to that site, called a guide RNA. Now, a lot of research has been going on in identifying very unusual DNA sequences in prokaryotes. Researchers were seeing these really mysterious repeating DNA sequences. What are they there? Why are they showing up? And it was actually a long time before it was recognized that this is an intrinsic kind of immune system that bacteria have, where they're capable of responding to the presence of foreign DNA that's been inserted by bacterial viruses. After it was appreciated the mechanism of this system, that this CRISPR was actually introducing double-stranded breaks into the DNA, researchers were able to take that and reprogram that for a wide variety of different genes within the cell, and to transfer this from prokaryotic systems to eukaryotic systems. And the reason that it's called CRISPR is that it stands for Clustered Regularly Interspaced Short Palindromic Repeat DNA Sequences. And this just goes back to the origin of these discoveries, where they were just finding these very unusual repeating DNA in these prokaryotic systems. So these discoveries really were the joining of two completely different research efforts. One of them was just looking at these CRISPR sequences. What are these repeating sequences doing in bacterial genomes? And the other was ongoing efforts in genome editing using things like these zinc fingers or other talons, these approaches here, which just weren't moving along very quickly. Once they realized what CRISPR was doing, they realized that they could come into this type of genome editing, grab this machinery, and then early demonstration of these sorts of site-specific genome engineering happened around 2013. So here's what the rudiments of the CRISPR-Cas system look like. So the CRISPR refers, once again, to these repeating DNA sequences, which are the sites at which these cuts can be made. And the Cas refers to the enzymatic machinery associated with this. In this case, Cas9 cuts DNA, and it cuts both strands to create a double-stranded break. Again, these breaks are introduced very specifically, and the specificity of the cleavage site is introduced by these RNAs, which are part of the CRISPR-Cas system. So you can introduce these into cells by putting all of these elements onto a plasmid DNA and expressing the system in a host cell. This will target double-stranded breaks at particular places, and then can either lead to recombination in such a way that you alter the gene and introduce a premature stop codon so that that gene is no longer expressed, or in more recent implementations, we bring in foreign DNA and actually edit that gene at that site. Yeah?
Does specific Cas9 sequences cut like variable amounts of nucleotides upstream from the PAM sequence? Because like they always say it's variable from like 3 to 4 base pairs, but they don't give like a specific amount. So if you were to have a really specific sequence, then and you don't want to lose a nucleotide, they like the papers never specify how many they cut upstream.
Right. You're talking about where exactly this cuts with regard with relationship to the PAM sequence? Yeah, I'm actually not sure whether or not they've been able to get good control over that. I don't know.
It's like completely variable then, like randomized?
I don't believe it's random, but I don't know how much control they have. I'll show you one example where they've been able to actually get really good control, and that's in cases where they don't introduce double-stranded breaks. Yeah.
Do you know why CRISPR is paused by the FDA?
Why it's paused by the FDA?
Yeah.
I'll talk about some of the regulatory issues. Yeah, I'll get to that in a second. So the initial system, CRISPR system, uses 2 different pieces of RNA, and one of the early improvements in this was just to create one conjoined guide RNA. That made the system a whole lot easier. And that improvement was immediately followed by these first few examples that this system could be used for in vivo genome editing. And here's the real bottom line. Researchers now had a flexible tool. They could move to any particular sequence, and they could easily guide the target to almost any location in the genome by simply designing a short guide RNA. Right? You just come in with a new little strip. This is not that difficult to make or to provide on a DNA plasmid, and you can direct these breaks to just about any point you want within the genome. And it was really those sorts of improvements that all of a sudden led to this huge adoption of this technology. So here are the number of publications year by year. Here's the projection for this year. Is going to be about 5,000 papers that will include the term CRISPR in the abstract. Does this remind anybody of anything? Really rapid adoption of a particular technology. I showed you one of these sorts of figures before. Yeah. Yeah, it was with PCR, which at first, for just a little bit, was like, okay, that's interesting, that's pretty cool. And then when they realized they could use thermostable polymerases, like, okay, anybody can do this, and it really took off. And so now with these introductions of these very convenient guide RNAs, this is really becoming adopted very quickly. So, right, here's just another example of how the system can be used for editing genes. This is the typical kind of implementation using this Cas9. And this Cas9, once again, introduces these double-stranded breaks into the targeted DNA. And then at that point, the juncture, the joining of these two strands in an imprecise way can lead to the introduction of a stop codon that prevents expression of that particular gene, or we can come in with an exogenous piece of DNA, insert it at that site, and have precise gene editing at that location. So what are the kinds of things that can be accomplished using this CRISPR-Cas system? We can correct genetic defects in whole animals. We can replace a mutated gene. We can alter DNA sequences in embryonic stem cells and then use those to, in culture to produce tissue that can be used to treat particular diseases. So somebody's got some type of heart disease, we can create cardiomyocytes. If there's faulty neurons, we can culture neurons, which can be used to as a therapy for neurodegenerative diseases. We can use this to create model systems for studying the genetic basis of disease. We can introduce particular human genes into, let's say, mouse models and study them in the laboratory. And then, very provocatively, we can change the DNA in the nuclei of reproductive cells that transmit information from one generation to the next. And so this is a big leap from what we've been talking about before, because this introduces genetic changes into an organism's germline. Yeah?
Is this DNA always like accessible though? Like, do they ever have to do anything to? No.
Yeah, so this gets back to this issue of what portions of the chromatin are accessible. And so I was just appreciating the fact that the efficiency of this type of modification can be related to some of the same issues about where nucleosomes are located, whether or not chromatin is condensed. You're right that the DNA has to be accessible, and that some of these same issues of like pioneer factors opening up chromatin and providing access to the DNA, this will be an issue for targeting particular DNA sequences in these therapies as well. So these sorts of changes to the germline will change the genetic makeup of every differentiated cell in an organism and allow that to be passed on to the progeny. So bottom line here, as pointed out by David Baltimore, is that changes to the human germline could be made using this simple technology. And it's, from what I gather, I haven't done it myself, it's not that difficult to learn, and it means that we can really change what the genetic composition is of the human species, which is kind of daunting. In addition to gene editing, we're beginning to figure out how to use the CRISPR system to do a lot of other really interesting things. So here's the typical way it's been implemented and what I've been describing so far, which is to have the CRISPR with the Cas9, which introduces the double-stranded breaks and allows us to do some kind of gene editing. But we can also use inactivated Cas. This would be called dCas9 right here. This does not have any nuclease activity, so it won't cut the target DNA, but it will bring this protein complex to a very specific site, again, guided by that guide RNA. Now we can attach other entities, functional groups, onto the CRISPR-Cas system. Like this might be a transcriptional effector, which could lead to upregulation of particular genes. We can think about having some type of epigenetic modifier, something that might introduce changes into histones or make methyl cytosine marks on neighboring DNA. We could put fluorescent groups onto an inactivated Cas and use it to image particular places in the chromatin and see how the chromatin might change in a dynamic sense as the cell undergoes different processes. Other approaches can be used to take the Cas and alter it so that it doesn't make a double-stranded cut, but just makes a single-stranded cut. And we would call that a nickase. And so we could begin to just make one cut at one particular site. This would be a little bit more of a delicate alteration at that target site. And that's been shown to be able to lead to base pair editing. And I'll show you an example of that as well. So yeah, these approaches for epigenome editing and base editing really indicate the fact that we can now fine-tune precise modifications onto DNA, even in a very complex genome. We're beginning to study the structural biology of this. We're learning about the organization of this protein complex, where the DNA and where the guide RNA are located within these complexes. This is one example of transcriptional activation. Okay? And so this takes an inactivated Cas and then uses the guide RNA to locate this protein complex at a particular region where we want to upregulate a gene. And then this uses this VP16, which is a very powerful transcriptional activator, to drive expression of that gene of interest. This was a really fantastic implementation of CRISPR-Cas technology that came out last year from the group of David Liu at Harvard, and this just blew me away. So what they've done here is that they've taken Cas9, they've used an altered version of it that has this nickase activity, so it's just going to introduce one particular cut in the target DNA, and then they've appended onto Cas an enzyme, deoxyadenosine deaminase, that will introduce this change from adenosine to inosine. which gets read as guanine by polymerases, and then after the polymerase replicates at that site, it changes an AT base pair to a GC base pair. Unbelievable precision, right? We're going to go into this really complex genome, identify one particular place that we want a modification to happen. We're going to direct this machinery to that particular site using this guide RNA, and by appending this enzyme onto it, we're going to say, okay, we want to make this one particular change at this one site, and we're just going to just tweak it just that little bit and potentially address deleterious mutation. So I think that this was a huge technical accomplishment, and I'm really in awe of what this group has been able to do with that. Companies are chomping at the bit to begin implementing this. Actually, some of the very first work on the CRISPR biology was done by a yogurt company in France, wanting to understand how bacteria that were used in culturing yogurt could protect themselves against viruses. So there's a lot of interest in biotechnology and using CRISPR-Cas genome editing to alter crop plants such as rice and wheat. Biology, obviously making particular changes into a whole bunch of model organisms that experiments are typically carried out on. And then in biomedicine, thinking about using this to alter stem cells for potential therapeutic benefit. The future, hard to say exactly. We could certainly use this type of approach to disrupt the expression of genes and viruses and perhaps protect us from viral pathogens. We might even be able to sterilize mosquitoes. There's been a lot of discussion about whether or not we could wipe out malaria just by making mosquitoes unable to reproduce using this type of genome editing. And then there's human gene therapy, which I think potentially could be the most exciting. As this technology was being developed, there were a lot of concerns precisely because of the power that was being established. And very early on, including at conferences in synthetic biology, many of the leading researchers at the time said, listen, there's serious risks associated with it. We all want to be able to reap therapeutic benefits, but we need to be cognizant of what could be done if we use this technology in an inappropriate way. And a general consensus was that we should not rush to alter the human germline with these sorts of technologies, that was going to be a sort of line in the sand that we weren't going to cross as far as developing new therapies. And Jennifer Doudna was very outspoken at that time, back in 2015, trying to get a real consensus of biologists that there would be a moratorium on editing the human genome itself. They said that they wanted to pause and evaluate its safety before they implemented these types of approaches. And there was a really interesting precedent for this because meetings were called back in the 1970s at the very introduction of genetic engineering and the use of restriction endonucleases to talk about what type of safety protocols needed to be established. And so that led to the whole use of biosafety levels, laboratories. You may be familiar with like BSL-2 or BSL-3 labs. All of those safety procedures were introduced at that time, and Doudna and her colleagues said, we need to get together and figure out how to make sure that we don't screw up the human species by altering our genetic material. Well, despite this call for a moratorium, just last month, bombshell news, a researcher has claimed to have created gene-edited twins. So this immediately drew outrage with scientists, bioethicists saying you shouldn't have done it too soon. It's ethically problematic. Some people said it was monstrous. I believe the Chinese government has also said that they are really concerned about the activity going on in this lab, and they're moving to try to control the advances that are going on in this particular lab. So what was the idea here? Does anybody know what this researcher was attempting to accomplish? Yeah.
From what I remember, he was trying to introduce a gene that could provide pre-negative HIV sachets.
Right. So he actually implemented this on a number of embryos, and the idea was to introduce the same type of changes into the genomes of these fertilized cells that would provide protection from HIV infection. And the idea is that once these embryos were born, that these would be individuals who would never have to worry about being infected with HIV and contracting AIDS. So that's kind of, a beneficial thing that these girls would not have to worry about it, but that's not the only way to protect somebody from getting HIV, right? There are other ways to prevent somebody from getting an infection. So that really, I think, was at the heart of a lot of the concern about what had been done. And a lot of the bioethicists and scientists said that if you're going to do something like this, there has to be a serious unmet medical need. The effort should be well monitored with sufficient follow-up, and there had to be informed consent of the parents. I think the parents did provide consent in this case, but this was not an issue. This was not an unmet medical need in the sense that there's no reason to think that these individuals would necessarily contract HIV if it were not for this intervention. Yeah.
Wasn't it specified in the contract that they would have, for the first 18 years of their lives, constant medical attention, and then afterwards they would sign a waiver to get free medical attention in the future as well?
Yeah, I don't know what the details were related to the agreement between the researcher and the parents in this case. I watched some of the videos from the researcher. Again, he's able to provide some type of justification, but it really doesn't address a lot of the concerns that had been expressed in advance. And it really also doesn't really meet the recommendations of the National Academy of Sciences that had met to evaluate when this type of intervention should be used. They said that we need to confine the use of gene editing in human embryos to settings where a clear unmet medical need exists. No other medical approach is a viable option. And that doesn't seem to be the case here. Yeah. Yeah, that's a really good question. Like, okay, there's these twins that have been born. You're not going to go ahead and do the experiment where you expose them to HIV. I guess that's not really the point, but that really does get at the issue of how do you carry out this science in a responsible way so that you can really demonstrate that you've done something beneficial. Just making these changes and saying, voila, I've done it, nobody else has been able to do it, in a way that doesn't advance the science and doesn't clearly benefit these individuals, I think is an area of real concern. So that's a big problem. I don't think there's any notion about how we're going to deal with this. Just because the National Academy of Sciences here got together and all these experts said, okay, we're only going to do it in these circumstances, it's not going to prevent anybody from doing this again in other settings. So how we're going to respond to this, I don't know. I haven't heard Jennifer Dowd not come up with any wonderful ideas about where to go from here. But, you know, that's kind of an issue. Like, people talk a lot about genetically modified organisms in our food supply. Now we have genetically modified humans to think about. But maybe that's not even the worst of it, because now we're coming up with such powerful techniques in synthetic biology that we may have the ability to create life forms that don't even really resemble life on Earth as we know it. Yeah?
Have they done any germline editing techniques with CRISPR in like animal models yet, or no?
There have been some tests in animal models, yeah. And, I mean, it works. Yeah, for sure. It's really not a debate about whether or not it can be done. It can be done. There's probably additional complications in introducing this technology into humans. That doesn't mean that it ought to be done. So here's Floyd Romsberg, who's at Scripps, had this phenomenal breakthrough a few years ago. I mentioned to you when we were talking about DNA structure that it doesn't seem as though base pairing, hydrogen bonding interaction is really crucial for base pairing. So Floyd Romsberg and his research group came up with this unnatural base pair, and now they've been able to show that it can be replicated. So that brings up the possibility that in addition to like an AT and GC base pair, that at some point somebody might be able to take and create an organism that has 6 different nucleotides in use of its genetic system. Whether or not that kind of organism would have a huge advantage over the rest of us pathetic entities that only have 4 letters in our genetic alphabet, I don't know. Floyd might be the kind of guy to try to find out. I'm not sure, but I'm just using this as one example of the types of advances that are coming about in synthetic biology, where we've got now so much ability to manipulate biochemical systems that we can drastically alter what it means to be a life form. And we need to figure out what is acceptable and what's not acceptable. And try to create some kind of consensus and controls before these sorts of technologies are let loose in an uncontrolled way. So beyond genetically modified organisms, we have this rise of synthetic biology. It's going to affect the things that you're going to be eating, for better or worse. I don't know. I'm very curious about that personally. But it's a reality, and it's going to also come to the clinic. And many of you will have to make these choices about whether or not you want to implement these sorts of therapies in any particular setting. As far as the food is concerned, I mean, that's a lot of what I think about. I believe in this adage, you are what you eat. And if we're creating these really unusual life forms and introducing them into our food systems, what the hell are we going to become when we eat these things? But for each of you here, especially those who are going to go into clinical practice, I want you just to consider how you're going to respond to the opportunities, the horrors, and the responsibilities of redefining what it means to be a healthy human. If somebody comes to you and says, I've always had my dream that my child would become a volleyball star, are you going to assist them in genetic manipulation of their progeny so that they can live out their dreams? Maybe a choice that you might end up having to make. So that's going to end it. I'll leave it on that sobering thought that we now have this incredible technology. You're going to be able to make those choices effectively because you're going to know what's happening on the molecular level within our bodies. So thanks again very much.